This is amazing. I never knew this could happen. Well, thank you. Yeah, there's a lot that I also learned that I didn't know was possible at this time point. Okay, how much of my work was based on previous work and how much is new? So there is no such thing as a completely new invention in chemistry. There's always a tiny bit of preliminary work. But for example, the core scaffolds, the blue part of the molecule that can break apart, that was known. The parts that binds to the protein was known. And that we can attach molecules to DNA was known. But I put these three things together in one concept. And the actual application of this to control expression of DNA, or actually the mRNA degradation process, those processes are actually completely new. Synthetic cells are also heavily researched topic. So making those is a standard procedure, but actually making them light controllable is something that not many people do. And then particularly what not many people do is actually embed them in an organogel, which I've done to like patent them, because that is something that's very specific to light, and no one has done this with blue light in particular. So there's a lot that I'm taking from the literature that is already existing in terms of methodology, like making the synthetic cells, but then, for example, embedding them in a gel and patterning them with blue light is completely new. So a lot of this work is very new. Yes, it is very exciting to work on new things. Thank you for the question. It is, that's one of the things I really like about doing chemistry. If you make a molecule that no one has ever made before, you are the only person who has ever held this specific molecule. You are the only person who knows how to make it until you publish your research. So that is something very humbling about chemistry in that way, that you are the only person in the world that currently holds this specific set of knowledge. So we got a question how far I can see my research going. And that is a very excellent question. I've already alluded to this being useful in terms of developing therapies. And I can see it going there to a certain extent. Probably not with blue light, definitely not with UV light, because those are not very like deep tissue penetrating. But people are working now on making them very much with a red light, and that actually gets quite far into tissue, so that might be useful as a therapy. There's people working on actually making DNA-based drugs way more accessible anyway. And so once that research picks up, we can actually bring our research into that as well. And that definitely is important. In addition, the work on synthetic cells actually is also picking up in recent times on that people now use them to interface them with real cells. So we've had Jeff in the group, for example, he worked on interfacing synthetic cells with bacteria and actually had them communicate with each other. And so there's people who now work on having synthetic cells communicate with human cells. Obviously at the moment in a flask, but hopefully in the future, we can actually use them to have them as like a skin graft that can respond to stimulus, for example, because they still operate like a real cell, and we can just incorporate more and more function. I got a question on if I'm working on this in my new job. Partially, let's put it this way. I will still work on photoactive molecules, and I will still work somewhat on biology, but I will leave DNA chemistry behind and work more on the fundamental processes of fluorescence, and I'll try to make molecules that can be used as very, very bright, like light emitters, particularly for things like fluorescence imaging in biology, so we can actually see biological systems, but also in terms of making new organic materials that can be used as organic LEDs, so OLEDs that are, for example, used in your phone screen. So similar area in using light to do things and using chemistry to make these molecules, but not on DNA. Will I be sad to leave this work behind? Did I want to move away from DNA? Yes and no. I really enjoyed working as part of this group, I really enjoyed working on these systems. The one thing that annoyed me about this work is how tiny everything was, and I was never able to actually see a lot unless I used a microscope. The amounts of molecule I used were not very convenient to handle, so that part I don't miss. DNA itself is a very fun molecule to work with, so that I will miss for sure. But I'm taking my favorite part with me, which is working with light. So what inspired me on going into this project was applying light and molecules that can interact with light on a system that actually has application. And so that part I'll be very glad to keep, because that is what really has been driving my research so far. How did I get to know about this area of chemistry using light? Actually through a lecture course in my third year on organic materials chemistry, where they talked about materials that can interact with light. And so that was when I first really learned about how these molecules can be used. I've always liked light. I've done a project actually in solid state chemistry during my undergrad in second year on using solid state materials that can interact with light, but using small organic molecules, particularly in a biological context, I learned about in a lecture course in third year. How much did I know about DNA before I started my PhD? Not a lot, to be honest. I never took the module we had on DNA chemistry in undergrad. I only had the most basic knowledge that was part of the core program. So one of the reasons why I actually chose to do this project was because I wanted to learn something completely new to me. And yeah, I didn't have to know a lot about it. A lot of it I could learn on the job, really. It is difficult, particularly coming from a pure chemistry background, having that much biology. But surprisingly, DNA is just like another molecule, and it follows the same rules as normal other molecules. So that wasn't too much of a shock, luckily. What was my favorite experiment? Oh, that's a difficult one. I would say anything that involves patterning of synthetic cells. It's the culmination of everything I've done. I'm using the DNA I've made, I've put them in synthetic cells, and then I show all the wonders that light has to offer in one single experiment. And I can actually visually see them because the synthetic cells are big enough to actually see with a microscope. Oh, thank you. Yeah, I'm glad you found it interesting. Sometimes I wonder if I'm doing this just for myself or if actually other people find it interesting.